Hi everyone. Today I'm going to be going over the sensory systems of smell and taste. So the learning objectives for this section is for olfaction, you need to be able to describe the underlying biochemistry. So you need to be able to describe in detail the biochemical steps that take place. And this can be found in chapter 34.1 in the online textbook. And the learning objective for taste is to be able to identify the mechanism of detection. We won't be going over in detail the individual biochemical steps for taste. And this can be found in chapter 34.2. Okay, so let's get started. Each primary sensory system consists of specialized sensory neurons, and these specialized sensory neurons transmit nerve impulses to the central nervous system where they get processed and are combined with additional information and yields a perception. Okay, so let's look at olfaction in more detail. What are odorants? Odorants are small organic molecules or compounds that can be carried as vapors into the nose. So small organic compounds. And what properties of odorants are responsible for odors? The shape of the molecule or odorant is what is important and responsible for odors. It is how these compounds interact with the binding surface of a protein receptor that is important. So shape of the odorant. And what kinds of receptors mediate olfaction? And this is something that we have already looked at previously in with cyclic AMP, GPCRs, and the phosphoinositide cascade. So likewise with olfaction, we are also going to be looking at GPCRs, and GPCRs are the receptors that mediate olfaction. Question 4 asks, what is G-olf? We know that with GPCRs, the G proteins contain alpha subunits, beta subunits, and gamma subunits. And we've always focused on activation of the GPCR is when the alpha subunit of the G protein is bound to GTP and releases the GDP that it was initially bound to in its inactive state. So G-olf is the G protein alpha subunit that is specific to olfactory cilia. Okay, so number 5, how do initial biochemical events transform into perceptions? So when an odorant molecule binds to its receptor on a neuronal surface, it is going to open up through a cascade of events, open up cyclic AMP gated ion channels, and the flow of ions through that channel is what is going to initiate an action potential, which is then transmitted to the central nervous system and yields a perception. So odorant binds, and OR is odorant receptor, which is the GPCR. And AP is an abbreviation for action potential. Okay, so the odorant binds to its receptor and through a cascade of events opens an ion channel, and the flow of ions results in membrane depolarization and generates an action potential. So now we're going to look at the specific biochemical steps in the olfactory signal transduction pathway. And this is the pathway, this set of steps is what you need to be able to describe for this test. So let's start first with the odorant binding to the receptor. So this will be step number 1. So odorant binds receptor. And binding of the odorant to the receptor activates the G protein. So what that means is initially, before the odorant binds, the alpha subunit of this G protein is bound to GDP. So right here. In this state, it is inactive. So when the odorant binds to its receptor, the GDP gets released from the alpha subunit and a GTP molecule is bound. So GDP released from GOLF. Remember, that's the alpha subunit specific to the GPCR in olfaction. So the GDP is released from GOLF and GTP binds the alpha subunit. Okay, so once that activation process happens, part of that is dissociating the beta and gamma subunits from the alpha subunit. So now the GTP bound alpha subunit or GOLF will bind to adenylate cyclase. So this is step number 3. So GOLF Okay, so the GTP-bound alpha subunit activates adenylate cyclase, and adenylate cyclase, as we know, is going to convert ATP to cyclic AMP and increase the intracellular cyclic AMP levels. So step number 4 is adenylate cyclase. Okay, and an increase in cyclic AMP levels activates the cyclic AMP gated ion channel. So step number 5 is right here, where the cyclic AMP increasing in intracellular levels of cyclic AMP is going to activate this cyclic AMP gated ion channel right here. And what that does is it's going to open up this ion channel, and these cations, which are positively charged ions, are going to flow through the channel and into the cell. So increasing cyclic AMP levels activates the cyclic AMP gated ion channel and allows cations or positively charged ions to flow into the cell. So the flow of cations into the cell depolarizes the membrane. So flow of Okay, so the flow of the sodium and calcium ions through this channel into the cell depolarizes the membrane and initiates an action potential. So let's quickly recap these steps in the olfactory signal transduction pathway. So the first step is the odorant binding to its receptor, the G protein coupled receptor, on a neuronal surface and activating the G protein. The G protein, when bound to GDP, is inactive. So odorant binding activates it by the alpha subunit releasing the GDP molecule and binding to a GTP. And the alpha subunit bound to GTP goes on to activate adenylate cyclase, which converts ATP to cyclic AMP. An increase in cyclic AMP levels in the cell is going to activate the cyclic AMP gated ion channel, and activating that allows for positively charged ions like sodium and calcium to flow into the cell. And when these ions flow into the cell, it is going to depolarize the membrane and initiate an action potential. And that action potential is then transmitted to the central nervous system and processed there, and then yields a perception of a specific odor. So for this test, you do need to be able to describe each of these biochemical steps in the correct order and understand how these steps go on to initiate an action potential that can then be processed by our brain and result in a perception or a physiological change. Okay, so now let's look at the sensory system of taste. Okay, so with the sensory system of taste, it uses a combination of different mechanisms to result or yield a perception of a specific taste. So the 5 primary tastes that can be perceived are bitter, sweet, sour, and salty, and umami. Okay. So now for the taste sensory system, you need to be able to identify the specific mechanism of detection for each of these 5 tastes. We will not be going over the specific biochemical steps, but just need to be able to identify what kind of a receptor or how these specific tastes can be detected, because they do have different mechanisms of detection. So with bitter, sweet, and umami, all 3 of these tastes, the mechanism of detection is GPCRs, or G protein coupled receptors. And for salty and sour, it's slightly different. So with salty tastes, the mechanism of detection is passage of sodium ions through ion channels. And for sour tastes, these tastes are detected by the effects of hydrogen ions on ion channels. So to quickly recap the taste sensory system, the 5 primary tastes that can be perceived are bitter, sweet, sour, salty, and umami. And for this test, we need to be able to identify the mechanism of detection for each of these 5 tastes. So bitter, sweet, and umami tastes are mediated by GPCRs, and salt is detected by the passage of sodium ions through ion channels, and sour tastes are detected by the effects of hydrogen ions on ion channels. So that is it for this section. If you have any questions, feel free to email me or write anything in the comments section, and I will answer those questions. Thank you.